Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome back to the Deep Dive. Today, we're looking into the brain of the nematode C. elegans. Tiny, but, famously mapped out.
That's right.
And here's maybe the most surprising thing we found. Scientists have known its complete wiring diagram, the whole connectome, for, over 25 years?
Yeah, decades now. All 302 neurons.
And this is the kicker, just knowing how it's wired, turns out that's not enough to predict what the worm will actually do.
Exactly. That's the core issue. You've got the map, the structure, but behavior is dynamic. It's function.
Right.
To really understand behavior, you need to see which neurons are firing, when, and how they coordinate. But C. elegans? Well, it's a challenge.
How so?
You've got 302 neurons, sure, but they're packed into this tiny space in the head ganglia, like incredibly dense.
Yeah.
Around 8,000 synapses in there.
Wow. So trying to image that, difficult.
Very difficult with standard volumetric methods. They're often too slow or can't resolve individual cells clearly in that dense environment.
Okay, so the mission then was basically build something new, something that could watch this whole 3D network firing cell by cell and fast enough to actually see the dynamics unfold.
That was the goal. Speed and resolution.
So how did they crack the speed part for getting those 3D images quickly? Let's start there. Figure 1, I think.
Yes, figure 1. They came up with a technique called Wide-Field Temporal Focusing, or WF-TeFo.
WF-TeFo. Okay.
It's a pretty clever two-photon microscopy approach.
Yeah.
Instead of scanning a single point of light across the sample, which is, slow.
The standard way. Yeah.
Right. They use optics to sculpt the light. They create a thin sheet or disk of excitation light across the whole field of view at once.
Ah, so it lights up a whole slice in one go.
Pretty much, about 1.9 micrometers thick. This means they don't need to scan side to side, the X and Y. They just scan axially up and down through the Z-axis.
That sounds way faster.
It is. It drastically reduces the mechanical scanning needed. They could capture 4 to 6 full 3D volumes per second.
4 to 6 per second. That's quick enough for worm neurons.
It's fast enough for the calcium dynamics they were tracking, which, while not instantaneous, are definitely the relevant timescale for C. elegans behavior.
Okay, speed. Check. But you mentioned density earlier. How do you actually tell one glowing neuron apart from its neighbor when they're all jammed together? That sounds like figure 2.
Exactly. That was the other big hurdle. And their solution was, really smart on the molecular side this time.
Not just hardware, then.
No, clever biology. They engineered a special calcium indicator protein, NLS, GCAM, and P5K. The NLS part is key nuclear localization signal.
Meaning
Meaning the fluorescent signal, the GCAM-P that lights up when calcium levels rise, is confined entirely to the neuron's nucleus.
Oh, I see. So instead of the whole cell body and all its tangled branches lighting up and blurring together.
Precisely. The signal comes just from the nucleus, a distinct spot for each cell. It prevents that overlap from the dense neuropil, the jumble of neuronal processes.
That's a neat trick. Did it work?
Yeah, they validated it carefully. They checked known neurons, like BAG and URX, which sends oxygen, and show this nuclear reporter capture their activity, both the fast changes and the slower ones, just like traditional cytoplasmic reporters would. So, reliable signal, but much better separation.
Okay, so now they have the tech. WF-TeFo for speed, NLS, GCAM-P for clarity. They put it together and recorded from, up to 70% of the head neurons?
In paralyzed worms, yes. A big chunk of the brain.
All right, so here's a thinking question for you listening. You're looking at maybe half the active brain of this worm, an animal with a known, quite structured connectome. What pattern do you expect to see? Neurons firing independently, randomly, or something more coordinated?
Well, the answer, looking at the results in figures 3 and 4, was definitely coordination. Massive coordination, actually.
Not random at all.
Far from it. They analyzed the correlations between neuron activity. They found distinct groups or clusters of neurons whose activity levels rose and fell together, strongly correlated, and other clusters were strongly anti-correlated.
Anti-correlated, meaning when one group is active, the other is quiet.
Exactly. And the largest cluster they found, It included key neurons involved in controlling movement, these premotor interneurons like AVA, AVE, AIB. Their activity was sustained.
Okay.
And crucially, this big cluster's activity was strongly anti-correlated with smaller clusters made up of head motor neurons, like RMED and RMD.
Wait, so the neurons planning movement are active when the neurons executing certain head movements are quiet, and vice versa.
That's what the pattern suggests. It mirrors the kind of push-pull, the antagonism you absolutely need for controlling movement, like switching between moving forward and backward.
That makes sense. Did they test it with actual stimuli?
Briefly, yes.
Yeah.
Figure 5. They combined the imaging with microfluidics to deliver controlled pus of different oxygen levels. And sure enough, the known oxygen-sensing neurons, BAG and URX, lit up exactly as expected. It showed the system works for tracking responses to sensory input, too.
So pulling it all together, what's the big innovation here? What does this platform really enable?
I'd say it's a truly enabling platform. It gives us, for the first time really, an unbiased look at the functional activity across most of the C. elegans brain at single-cell level, fast enough to see dynamics. It's the functional map that perfectly complements that anatomical connectome we've had for so long.
So we move from just the who's connected to who diagram
To actually watching how the system works together, how these widespread patterns generate behavior. It's a big step.
and the tech itself, can it be used elsewhere?
Definitely. The optical setup is quite flexible. You can combine it with other things like stimulating specific neurons with light, optogenetics, or even electrophysiology. And because it's good for dense tissues, it could potentially be adapted for other small model systems, fruit fly larvae, maybe zebrafish brains.
Okay, let's recap the main takeaways then. 3 key points.
Sure. First, the WF-TeFo imaging. Using that sculpted light avoids slow point scanning, allowing fast 3D imaging.
Got it. Second.
Second, the NLS-G CAN B5K reporter. Putting the signal just in the nucleus was the crucial biological trick to tell apart those densely packed neurons.
Makes sense. And third.
The big finding, behavior, doesn't seem to be just isolated neurons firing. It looks like it emerges from these large-scale, coordinated, often antagonistic patterns of activity across big chunks of the brain.
Yeah.
System-wide correlation.
Right. Now, quickly, is there any limitation we should keep in mind?
Well, yes. Because the signal's only in the nucleus, you don't see the really fine-grained calcium signals happening out in the dendrites and axons and neuropil. So, detailed studies of processing within a single neuron's branches still need other methods.
Okay, fair enough. But it leaves us with a pretty profound thought, doesn't it? If even this relatively simple worm brain seems to rely on such widespread, coordinated activity for its basic behaviors, how much of our own complex thoughts and actions might be driven by similar, massive, system-wide correlations that we just haven't been able to see yet? Something to think about. Until next time on Deep Dive.